just wanted to thank everyone for showing up to this Health AI seminar today. Our speaker is Wouter Meuleman from the Altius Institute for Biomedical Sciences in Seattle, Washington, where he's been a group leader there for about 5 years. Prior to that, he was a postdoc with Manolis Kellis, working on large-scale projects involving the Roadmap Epigenomics project and other things, and is interested in artificial intelligence and its use for augmenting other aspects of genomic science. And recently he was awarded an R35 as one of the NHGRI genomic innovators from last year. So yeah, we're very happy to have you speak today, and the floor is all yours.
Thanks. Thank you very much for that nice introduction. Thank you for having me today. I'm excited to present our work. Let's see, let me share my screen here. All right. I will make sure to leave a couple moments for questions midway through the talk, but feel free to also put questions in the chat if convenient. So yes, my name is Wouter Meuleman. I'm a principal investigator at Altius. Altius Institute is a nonprofit research institute in Seattle, Washington. The main focus of the institute is to do academic research. We're funded by public funding, including NIH, and as well as private funding. So today I'd like to talk to you about our work on mapping and navigating the human regulatory genome. So as a bit of an introduction, let's look at the human genome here. This year marks the 20-year anniversary of the Human Genome Sequencing Project, when the map was completed, although I guess technically the map was really only actually completed a few months ago of this year. In 2001, this initial map was published. It was kind of a big deal, and it's something that many of us, certainly myself, still benefit from to this day. However, if you look at what was actually published, there's a lot of information on a very sort of uncharted territory. But if you look at the actual map, it's essentially contained in 2 PDFs in the supplementary material of this Landner et al. Paper in Nature. And one thing you can do is you can print this map out and look at it. There were not really very many other ways to actually consider the totality of this map. In fact, if you think about how you would navigate a map like this at the time, it sort of felt a little bit like going on a road trip somewhere in Europe, for instance, and having an old-fashioned map on the spread out on the hood of your car. And so while the map was in 2001, the navigation system to use that map kind of felt like it was from 1981, 20 years prior to that. Now, if we fast forward to today, surely things will be different, and they are. In 2021, we have much better maps. We have much more data sets, higher resolution, more granularity between individuals, between cell types, etc. So our maps are much better, and they've moved from paper to screen. There's many advanced and less advanced browsers out there for people to start browse their favorite regions. But that's about it. You browse your favorite regions, you look at your favorite annotations. There's not a whole lot of navigation beyond that. And so while the navigation systems have improved by way of these browsers and other rudimentary query systems, the navigation system is still kind of lagging about 20 years behind. And so these very valuable data sets, these genomic data sets, these maps, are still to this day very hard to access and particularly interpret at scale. So let's think about maps for a second. What would an ideal genomic map look like? And in my view, an ideal map looks like this. This is a map of the Walt Disney World Resort. I have never been here. However, when I look at this map, even as an adult, I immediately become curious about what's going on here. What's this castle? What's going on here? What's this big sphere? What are these buildings over here? They elicit a sort of sense of wonder and sort of a desire to start exploring this space. But of course, this map is completely wrong. This map is wrong on so many levels, literally. It highlights certain things that are deemed to be relevant, but at the same time, it hides a ton of stuff that is maybe less relevant at this scale, like how long are the lines to get into the Magic Kingdom? Where are the bathrooms? When was the last time the grass was mowed, etc. And so I would argue that this map, in my opinion, is the perfect map of the Walt Disney Resort, mostly because it's wrong. It's useful and it's great because it's wrong, because it, by definition, needs to summarize reality and highlighting only the most quote-unquote relevant information. And so what is considered relevant, of course, is quite subjective. It really depends on what the subject is that you're mapping, what the resolution of the map is, what data goes into it, and particularly the context. If I were to be interested in maintenance of the Disney Resort, I would like to see where was the last time the grass was mowed, for instance. And so if we think about building maps for genomics, and particularly trying to get to this, quote unquote ideal of a Disney map for genomics, then we need to start finding and annotating the most relevant parts of the genome, or in other words, the most exciting attractions. And so that's what I'd like to talk to you about today. In search of relevance, annotating the regulatory human genome. First, a little bit of background here. As most of you will know, the organization of the human genome is very complex, both from a spatial as well as an operational point of view. The majority of the genome is actually non-coding. That means it doesn't code for proteins. And there are many sort of regulatory elements hidden in this non-coding space. It turns out that the majority of disease-associated genetic variation, such as found using genome-wide association studies, maps to these non-coding regions and are strongly enriched in regulatory elements. And so that means we need to really try hard to understand this regulatory genome better, and not just the protein coding part. And so luckily, there have been a plethora of experimental genome-wide assays that developed over the last few decades that allow you to sort of map this non-coding genome in great detail. Unfortunately, these maps at a case-by-case basis can be very hard to interpret. Today, I'm going to argue that if you have many of them, you can actually learn a lot. So over the years, as Chris alluded to in the introduction, I've worked on various aspects of mapping this non-coding human genome. During my PhD, I worked on chromatin domains, and in particular, large megabase domains that line the inner rim of the nucleus, called lamina-associated domains. And these things are strongly associated with repressive heterochromatin, so a repression of genes that are contained in there as well. For my postdoc at MIT with Noel Skellis, I started looking at chromatin states and starting to map chromatin states by way of new computational methods. And after that, I had the opportunity to join the Altius Institute as a principal investigator, where I started shifting my focus towards chromatin accessibility, which is essentially trying to find single elements of accessible chromatin that are often indicative of individual DNA binding sites. So I'm going to focus today on chromatin states and chromatin accessibility. And I'm going to focus on these things to show you two types of genomic annotations that I think are relevant. One is based on chromatin states, which tell you something about what type of functionality does a genomic region encode. For instance, is a piece of DNA representing a promoter, the start of a gene, or does it indicate that a gene is being transcribed or repressed, etc. And then largely orthogonal to that, chromatin accessibility, which doesn't necessarily tell you anything about the function of the piece of DNA, but it does give you a lot of information on where, in this case, the human body, a piece of regulatory DNA might be utilized, for instance, in a heart or in brain or immune system. And let's focus on the first section here. If we look at the chromatin structure, or the way DNA is organized in the cell nucleus, probably all aware that that's by way of chromatin, and which is essentially DNA tightly wrapped around these histone octamers forming nucleosomal structures. Now these histones have tails that can have little chemical modifications applied to them, which we refer to as epigenomic marks today. It turns out, through decades of study, that these epigenomic marks are associated with certain functional elements. So it turns out that if you see a lot of a particular type of modifications, and I'm just going to refer to this by way of their sort of shorthand notation, for instance, H3K4 trimethylation, that's a good indication that that particular position, that particular piece of DNA acts as a promoter to a gene. Similar for enhancers, transcribed regions, repressed regions, etc. And so we can exploit this if we can measure where all these different marks are across the genome in a particular sample of interest. Then we can exploit this to find where these potential different functional elements are. And so this is something that the NIH Roadmap Epigenomics project set out to do a while ago, to basically create reference maps of the epigenome rather than reference maps of just the human genome. This was a multi-center effort across the US and as well as Canada. And I was at the time at MIT, where I was leading the integrative analysis of all those data. And so to give you a little bit of a flavor of what that would look like, at the time we were primarily interested in ChIP-seq, which is chromatin immunoprecipitation sequencing, which essentially allows you to find where these epigenomic marks are across the genome. And so here I'm showing you kind of a cartoonish representation of some of these data, where you have 5 different of these epigenomic marks. And a particular genomic location might look like this in terms of how these marks are distributed across that locus. This is informative in and of itself, but it also becomes quite clear that there are certain patterns here. There are certain marks that like to co-occur. There are certain marks that really do not like to co-occur. There's areas where there's nothing. There's areas where there's a lot of stuff going on. In other words, it's a complex picture. And particularly if you start collecting this across multiple cell types, in case of Roadmap, more than 100 different cell types and samples, it starts to become quite a complex data set across the entire genome and across many epigenomic marks. And so you need computational methods to make sense of some of these data, or at least I would highly advise you to use those. This is why we applied a multivariate hidden Markov model to basically essentially try to find out what are some of the prevailing patterns in these epigenomic mark ChIP-seq tracks that sort of recur across the genome, and can we capture those in a very limited number of discrete states. So for instance, here there is nothing observed, so which means that you have certain white state. For instance, here you mostly see H3K9 trimethylation resulting in this purple state. You may also see combinations of certain marks. Here we see K4 monomethylation with K27 trimethylation, and that results in a separate sort of hybrid state. And so we can train this model based on all the data that we have, and essentially get an idea of, these are the states, at first unlabeled. These are the states, and these are the combinations that sort of make up that state. So if you see an area that is red, then you can essentially look up, okay, well, that was mostly driven by the presence of K4 trimethylation. Now, at this point, these are just states, just 50, in this case, 15 states. We can assign them a color, but then we can actually go back into the literature, as well as look at how these things are distributed across the genome to attach tentative labels to them, to try and interpret these states. It's a very important part of this, because you want your summarized model to be more interpretable than the original raw data. And so, for instance, for this first state, we find that it's mostly driven by K4 trimethylation, and we know from literature that that is a sort of a canonical mark for promoter regions, and so this state is marked as a transcription start site state. Importantly, we can then roughly classify all these things into these sort of separate categories. And importantly, we note that some of these are much more prevalent than others. For instance, about 2/3 of the genome is in this quiescent state on average, whereas promoters are exceedingly rare. Now, this kind of model trained on all these data now allows us to take this large data cube and essentially reduce the dimensionality to only 2 dimensions, which is much more manageable. And so if you see a color here, like this red streak, for instance, it corresponds to state here. So given a map like this, you can essentially look up for any region of interest, any of your favorite regions in the genome. How does it behave in terms of potential function or functional role across this wide range of cell types? This turned out to be a very valuable resource for a lot of people, and has been used by us and many others to predict regulators of regulatory elements and genes using motif enrichment, to predict links between genes and the possible enhancers that might regulate them, and also even to link traits to their relevant tissue types by way of using GWAS data. I see one question here in the chat by Srinivas. Have you included H3K27 dimethylation in the state mapping, as that seems to be most abundant state of H3K27 in many cells? That's a good question. So in this case, no. In this case, we have only looked at H3K27 trimethylation. At the time, that was considered to be sort of the the most clear mark for essentially bivalency in these samples. There are other models we have that are more extensive, have more states as well as more input marks, and these might include H3K27 acetylation for finding active enhancer regions, but not H3K27 dimethylation to my knowledge. All right, so given this resource now, although this is hopefully useful for many people, it still doesn't quite solve the problem of navigating these maps. In particular, the genome is very large. I'm showing here a very, very small portion of the genome. And of course, the number of cell types or samples is ever increasing. Right? And so at the time, this was around 100 samples. And by now, it's easy to put together a resource like this for about 1,000 or more samples, as we've done recently. And so we need better methods to find interesting areas in maps like this. And so this is when we started looking at, how can we develop that? And we realized that if you look at a small portion of the genome and across a lot of cell types, then essentially these chromatin states, where so every row here is a different cell type, this is a small portion of the genome, every row here can essentially be considered a sequence of colors. And so in a way, looking at it like this is sort of a multiple alignment of sequences. And so if we talk about multiple alignment of sequences, such as here shown for various variations of the c-MYC binding site, we never talk about the c-MYC binding site like this. We always talk about it like this in terms of sequence logos. And so there's some fairly simple information theory behind converting a multiple sequence alignment like this into a logo like this. And so that's exactly a similar approach that we use for converting, for trying to interpret and navigate these chromatin state data sets, except now we don't use nucleotides, we use colors. And so let me make it a little bit more concrete for you, give you an example of what that looks like, squash this view a little bit. And so this is a small locus on the human genome, and if we apply exactly the same trick as we would use to obtain a sequence logo from a multiple alignment of sequences, if we apply that to this data set, then it turns into something like this, which we refer to as Epilogues. Now, what does this mean? The y-axis here basically shows you the amount of information content, or saliency, as we call it. It tells you how surprising it is to see a particular combination of chromatin states at a given location in the genome. So here we see, if we zoom in a little bit, we see that this area, this light green area, scores very high, and that is because this particular state is exceedingly rare, and at this particular position, there's a lot of it. Likewise, it'll essentially downplay some of the areas that are really seemingly not very interesting compared to what you would expect by random chance, namely these sort of white intervening areas. And so areas like these putative enhancer areas here readily pop out of the data rather than sort of be scattered in sort of a sea of white. And so this basically shows you that these saliency metrics highlight relevant information. Again, we are in search of relevance here. And to make that point a little bit stronger here, we started looking at whether the height or the extent of the saliency levels tracks with anything we care about. And it turns out it does. It turns out the higher the saliency level, the stronger in general sequence conservation is of underlying sequences. The the more trait and disease-associated variation we find there, as well as the more regulatory sequence content we identify there by way of transcription factor binding motifs. And so, in a way, these saliency metrics can be seen as, one method to finding the most exciting attractions in this case, these data sets. I think this is a good time to stretch a little bit while I show you a small demonstration of what that looks like in a browser we developed for this. I hope that came through, but this was essentially a summarization of about 5,000 genome-wide ChIP-seq data sets. I invite you to visit that browser and check it out for yourself. So what can we do with this? Well, if we can measure relevance, or we measure saliency across the genome for a large group of samples, that means we can do the same for subsets of samples, right? We can do the same for group A versus group B, and then compare the two and see if there's any differential signal. And that's exactly what I'm showing you here. So what we did here is we built Epilogues for, in this case, embryonic stem cell samples, and as well as anything else really as part of roadmap. And then we can systematically, for every position in the genome, find if there's any differential signal between the two groups. So here I'm showing you a locus BTG2 that shows a very strong difference between ES cells and non-ES cells. In particular, a lot of these marks, the majority of the marks shown in the top track here, are all sort of repressive, meaning that this locus is sort of currently being repressed in the ES cells. But in other cell types, very clearly, the majority of the marks here are actually corresponding to active marks, so active promoters and active transcription and things like that. This is in line with what you would hope to see because BTG2 is an anti-proliferation gene, and so it's actually sort of silenced and repressed in ES cells, which tend to proliferate more than others. This is one example. Another example that I think is striking is when you don't compare groups of cell types, but you instead just completely disregarding any information on cell type, you just take all material of male donors versus female donors, and you follow the same procedure. You get Epilogues for male and Epilogues for female donors. And immediately you'll see that there's essentially 3 loci that show strong differential scores. First, there's the canonical exist locus, which plays a major role in axon activation in females. And indeed, you see that there's essentially only activity in females and not in males. And the same holds for two other very related loci, namely the fire and ice loci, where you see a very strong readout in terms of differential signal between male and female. I'm showing you this example because Epilogues doesn't just tell you which loci show a different epigenomic signal, but it immediately tells you why. So in that sense, it shows you relevant loci and offers you an initial interpretation on why these regions are different or what drives the differences. And so to summarize this part on chromatin states in Epilogues, we basically devised this method to go from a large unwieldy data cube like this to a more manageable 2D matrix, and then further to a almost 1D representation, with the idea that we can reduce the dimensionality, increase interpretation, while hopefully not reducing the information content or the actual information contained in it too much. The software is freely available, and again, I invite you to browse around the genome in a way that you may not have done before. This is a good place to stop to see if there's any questions on this first part. Of course, also happy to wait till the end. Okay. I'll continue on to the next step, chromatin accessibility. So this is where we are interested in finding out where, in this case, the human body or which organ system, potential regulatory elements are being utilized. And so to go back to this illustration again, we're now moving to the right a little bit, where we have DNA binding proteins able to access and bind to sort of naked DNA or accessible DNA, which means that there is no nucleosomal structure protecting the DNA from being bound. We can, and so that is indicative of potentially being a regulatory element. We can identify these types of regions by using an endonuclease such as DNase, which cleaves naked DNA. And then you obtain these fragments and you use paradigm sequencing and map them to the genome. There have been other methods developed like this to do this. For instance, ATAC-seq. We use DNA-seq, which is sort of the gold standard for doing these kind of chromatin accessibility assays. And so what happens if you do this, you obtain a lot of these short fragments, you sequence them, map them to the genome, and you'll find that there are areas of high enrichment. So a lot of fragments map to this locus or this region, and not many fragments map over here. And so this tells you that if there's a lot of cleavages at this position, it means that that DNase in general there was highly accessible. And so these data sets are extremely rich for finding, for identifying regulatory elements. But again, on a case-by-case basis, they might be hard to interpret. And so in order to attempt to make a map of these DNase1 elements, these accessible elements, we did this as part of the ENCODE project for more than 400 different cell types and states, spanning more than 700 different biosamples across the human body. And I'm showing you here a small sampling of just the hematopoietic system. As you can see, there's a wide range of different cell types. And you also see that that corresponds to a wide range of different patterns with which these accessible elements occur. For instance, there are elements that seem to be quite or nearly constitutive across all cell types, others that are really restricted to a certain compartment only, and others that seem very cell type specific. And so in order to start making sense of these, in order to try and consider these onto the same coordinate system, we devised a very simple method to delineate what we call consensus DHSs, or consensus DNase1 hypersensitive sites. So I'm showing you a small cartoon example of this here, which is actual data, but just for a small set of samples. In this case, we find accessible elements in all these cell types. And what we can do is we can essentially detect the peaks on it, project them down onto the genomic axis, and delineate these so-called consensus elements, which are made up of a start position, an end position, an estimated centroid or center of mass, as well as a core region, which tells you something about what the positional stability of those elements is across those samples. This way, we're able to identify more than 3.5 million of these consensus elements in the human genome. And we annotate them also with a unique identifier, which has a number of interesting properties that I'm happy to talk to offline. This method is very simple, but it works very well across the range of positional stability you could see. So for instance, here we have a bunch of accessibility events that are less well behaved, as the first example I showed. Namely, these seem to be moving around a little bit. There's a slight skew in it. And so our approach is still able to nicely identify these and sort of annotate them, just with less confidence on the exact positioning of the center. Similarly, because we're using this approach and we're identifying individual accessibility events in individual samples, we're able to also identify sort of atomic accessibility events that are very close to each other. And so what this now allows us to do, now that we have this index of DHSs across different cell types, is we can build a matrix like this, where you basically look at, for each of these 3.5 million DHSs, what is their behavior across the 700 and something data sets we have? In this case, I simplified it by saying, is an element accessible in a cell type, yes or no? So if you order those columns a little bit, then you'll start seeing this pattern emerge. Naturally, you expect to see a strong diagonal here. Similar data sets will likely share similar repertoires of DHSs, such as the case here with T cells and blood and muscle and brain, etc. But we also know that there's a lot of off-diagonal behavior. In this case, for instance, there's tens of thousands of DHSs that, although they show up in lung tissue, they also show up in various other samples here. And so this means that if we are looking to annotate these 3.5 million DHSs, or say something about what their, what their properties would be, we cannot just apply something like we've done for roadmap, which is a simple clustering of the columns here to find subsets or modules or groups of elements. And so instead, what we decided to do is we decided to take this matrix and do a so-called decomposition. What that means is we use non-negative matrix factorization to essentially reduce this matrix, the dimensionality of this matrix, to something, instead of 733 data sets, to 16 components, shown by 16 colors here. So the most important point to take home here is that each DHS, each of these 3.5 million DHSs, is now described not in terms of 733 data sets, but in terms of a mixture of 16 components. To make that a little bit more concrete here, I'm showing you 10 examples here. So for instance, if you have some DHSs might have primarily an individual component, others might be a mixture of two, or even a mixture of all or most of the components. So what does that mean? What does that actually reflect? It turns out that those components very nicely align with sort of distinct biological contexts. For instance, if we look at this primarily yellow DHS, we see that it shows up consistently in trophoblast or placental tissue, and not so much in other cell types. If we look at mixtures, for instance, here orange and red, we see that it shows up in CD8 T cells as well as CD34 positive cells, but in not much else. And on the other end of the spectrum, these sort of rainbow DHSs, they indeed show up across all or most cell types. What I'm showing you here is for each of these components, I'm showing you the most strongly associated data sets. So these are the data sets that have most of the yellow component associated with them. And this allows us to give tentative labels to these components, right? Just like with the chromatin states I've shown before, the colors are nice. There's 16 of them. It's manageable. But if you don't know what the colors mean, it's not that useful. And so if we look at what the top data sets are, not just the top one, but the top 15 or 20 or 30, we see that a very, very consistent pattern emerges, and that basically the top 15 of the yellow component are all trophoblast or placental related. So the top 15 of the green components are all cardiac related, etc. And so this, together with a motif enrichment analysis on DHSs that are primarily in one component, which resolves a lot of the well-known regulatory factors for these kind of cellular contexts, gave us confidence to assign these probably imperfect, but hopefully useful labels to each of these components. So what this gives us right now is a DHS index of accessible elements with a start, end position, centroid, as well as an indication of positional stability and a unique identifier. We find more than 3.5 million of them. And for each of those, we can also assign them to one, in this case, majority component, or even the full mixture of components. But I'm going to keep things relatively simple today and assign every DHS to a single component. So now we've basically distilled the 733 DNA-seq data sets into what you see on this page. And so now the question is, how can you use these annotations to further identify regions of quote-unquote relevance in the genome? We have a separate browser for these data as well that I invite you to take a look at, as well as all the code is available for doing this on your own data if you'd like. So one thing we saw as we started browsing through the genome with our browser is that there is a lot of clustering of similarly annotated DHSs across the genome, and particularly around genes. So for instance, for this cartoon example, if you have a certain gene of interest and you look in and around it, it turns out that there's very often you see similarly colored DHSs. And so what we can do is we can do a very simple test for overrepresentation of any given color or component in and around the gene body. And if that's significant, we can assign a label. We can assign that label to that gene. Let me show you an example of that. For instance, here we have the GATA1 gene, a well-known erythroid factor, and we see that in and around this gene, there's an abundance of red DHSs associated with the myeloid and erythroid component. Since the erythroid, since GATA1 is a very well-known erythroid factor, there seems to be a link between sort of the characteristics of the regulatory landscape in and around that gene and the potential function of that gene. Same holds for a bunch of other examples I'm showing you here, like FOXP3, which has a lot of lymphoid-associated DHSs in and around its gene body. In fact, we did this for all genes in GenCode, and we find that we're able to annotate more than half of the protein coding genes to one or sometimes multiple components. And that these annotations are very typically in line with sort of the known function of these genes. Like, for instance, here in the lymphoid component, we identify a lot of lymphoid and immune-related genes, as well as here for the myeloid and erythroid component, the stromal component, etc. Particularly also for transcription factors. If we only focus on transcription factors, for instance, here for the cardiac component, the top five transcription factor genes identified in this way to be associated with the cardiac component are essentially all the main cardiomyocytes or lineage specifiers. And so this is another example of how we can use these DHS component annotations to filter for relevance in the genome. You now have a gene, and you can look up, what are the putative regulators for that given gene, at least in a local vicinity. Beyond gene annotations, we also looked at whether these DHS components seem to preferentially contain heritability signal, as found by genome-wide association studies. It's long been known that DNA's hypersensitive sites strongly enrich for these type of relevant genetic variation. But we started looking at if you specifically partition heritability in terms of these DHS components for relevant traits, whether that gets even stronger. And that's clearly the case for many traits. So for instance, here for reticulocyte count, we find that there is a very strong enrichment of capturing genetic heritability in DHSs specifically marked with this myeloid and erythroid component. And interestingly, this is much stronger than any of the 85 or so genome-wide annotations that people typically use to partition heritability, such as protein coding regions and promoters, etc. And this holds this strong enrichment of trait heritability inside these DHS components holds for more than 250 traits as found in the UK Biobank. Again, another example of how these DHS annotations allow you to essentially put another lens on the genome and essentially filter a data set to hopefully drill down to more relevant subsets of your data. So I'll pause there for a second to see if there's any questions on that second part. Okay, so then, by now, oh, I see that there's one question. Okay, so the question is for part one about chromatin states and Epilogues. For the 3D to 1D transformation, how do you mathematically guarantee the information content loss is not much? I think the short answer is we don't. We don't guarantee that the information content loss is not much, but when we look at large chromatin state data sets, we are typically interested in summarizing the sort of the high level patterns while taking into account the rarity of the events, right? So if you let me put it different. So it's not just an average of these chromatin states across many different tissue types or cell types. We take into account what the expected rate of a particular chromatin state is. And so if a particular chromatin state is fairly rare, but then occurs in only a few of the 100 and something cell types, it'll very likely still pop up as being sort of the consensus chromatin state after that transformation. So while we do not mathematically guarantee that the information content loss is not much, we set out specifically to develop this method for highlighting the higher level patterns. If you want to find out specifics on particular subsets of data sets, you would have to essentially run the same methods, but on a subset of samples of interest. I hope I somewhat answered your question. Then a question from Larry Hunter: how did you pick the dimensionality of your NMF? Are the 16 states fairly pure? That's, I would almost say, a classic question. It's a very good question. It all depends on, I think it's a trade-off between how much of the biological complexity you want to explain versus how interpretable you want your model to be, right? And so what we did is we actually, we tried a whole range of components for this NMF. And what we did is for every time we looked at the reconstruction error of the original matrix, and we sort of plotted essentially the F1 score. And then we started seeing, we basically found the point in which we sort of had like diminishing returns, at which point, if you would add more components, you wouldn't really add that much, right? Then we also looked at the interpretation of the components. So we looked at, adding a new component, does that give us confidence that that component is capturing something meaningful? For instance, if we would have gone for 15 components, it turns out that we would not have differentiated between myeloid and lymphoid. So we just have like one sort of hematopoietic state or something, right? And so we thought that that additional step was a relevant thing. But yeah, it's a great question. It is, if you would use a purely objective measure, some sort of objective measure for finding out the ideal number of states, you would end up, or components, you would likely end up with such a high number that totally eclipses the utility of these models. And there's a question from Joanna Rankenberg for slide 32. When you look at the centroid for DNase activity, do you also match the centroid position with sequence homology or just sequence position? I'm not sure I understand that question. So the DNase activity is mapped essentially to the same reference genome. And so what we do is we identify peaks in individual biosamples, and then we estimate what the summit position is in individual samples. We project those down onto the genomic axis, and sort of essentially the average or the median position of all those represents our centroid. We don't take into account the underlying DNA sequence. If that doesn't answer your question, then please shoot me a note afterwards, and we can discuss. So maybe you've gotten a little impatient by now, and you're saying like, well, you're showing me these methods that you're using to build annotations to find relevant regions in the genome, but where is my Disney map? Show me my Disney map for genomics in particular. And so I want to spend the last 5 minutes or so thinking about that. So if we look at the genome, it's, of course, we typically show this as a 1D string in the genome browser or in your printed out version of the human genome. And in order to get something that we can work with on the screen, we could, it's better to have this in 2 dimensions. And so we can use something called space filling curves to start progressively folding up this 1D genome into 2 dimensions. So we can use, for instance, Hilbert curves with increasing orders to get more and more granularity and sort of higher resolution individual elements. We can keep doing this until whatever we want, until essentially we get to a resolution that we like, and we can fit the entire human genome in a 2D space such as this. This has been done by other people before for various purposes, and it has some really nice properties for looking at genomic data. For instance, regions that are close in 1D are guaranteed to be close in 2D as well. Something that's close in the linear genome is going to be close in this space filling curves. The genome, so the genome coordinates, the reference genome provides a fixed scaffold. So you have your coordinate system. This is purely based on coordinate system, not based on any kind of annotations or data. And so you can take any data, and as long as it maps to the genome, it maps to this sort of 2D scaffold. And the nice thing is that it has full coverage by definition across the genome. And if you want a higher resolution, you just increase the order of the Hilbert curve and you can sort of sharpen your view. So that means that we can take the annotations that I was just talking about, fold them up in a similar way. Maybe we combine them across different types of annotations, like a functional annotation, a topological annotation, and contextual annotation. We can have whatever, as long as it maps to the genome, we can map it onto this. And so what I'm showing you here, without going into details on how exactly this was derived, is an integrative annotation covering about 6,000 genome-wide data sets. And the reason I'm showing you this is, like, even though all these genome browsers seem, they seem hard to access, they don't feel natural to start browsing. They don't give you that same sort of sense of wonder that maybe a map of Tuscany, or in my case, the Walt Disney Resort map gives you. The point I would like to make with showing you this is, like, I think given the right metrics for distilling sort of relevance in the genome, I think we are, at least conceptually, really not that far off from having maps that could invoke that similar kind of sense of wonder, and sort of can actually encourage exploration. This is, of course, a little bit of a tongue-in-cheek depiction. I don't actually think that the genomics field is going to move to browsing maps like this anytime soon, but I do think it illustrates an important point, that if I look at a map like this as a genomicist, I see a lot of structure, and I'm very much interested why this region looks so qualitatively and quantitatively different from this area over here. What does that mean? And so I think there is a great opportunity for using maps like this, or at least formats like this, to allow users to look at data, or summarized data, and ask questions like, if I'm interested in a particular region in the genome, identify other related relevant regions, right? And the way this has to be done is not through just this summarized landscape. I think there is an enormous opportunity for actually showing only that summarized view to a user, while in the back end, of course, there's a machine-assisted solution that has access to essentially all of the data that ever went into that given annotation. The nice thing is that this is applicable to any kind of genomic data set, as long as it's mappable to the human genome, which nowadays also includes transcription activity, which has been mapped to single base pair level, conservation, et cetera, et cetera. And so I don't think it becomes a matter of man versus machine at this point. I think it's more a matter of man with machine, where a human makes decisions in this landscape, and it's augmented by machines in the backend providing full data-driven guidance. That's a little bit of a taste of where I think things are going. And so, to summarize, I think we have great maps. We can make them, I think, even better by what I showed you today, but we definitely need better navigation systems. And I actually would like to make a bold statement that these kind of efforts are, I think, part of a new field that I call augmented genomics, where genome scientists are not replaced, but supplemented by machine intelligence that has sort of a full access to all of the data behind it. This is work by a ton of people, not in the least the people in my group, and those who have gone on to do bigger and better things. I can thank NHGRI for funding. And all of you for your attendance and questions.
Thanks so much for what a great talk. Still have time for some questions.
So there's one question from Debashis Kosh. Aren't there other types of space filling curves from 1D to 2D? More generally, since humans process information in 3D, wouldn't 1D to 3D make sense too? Very interesting question. So as for the first part, are there other space filling curves from 1D to 2D? Yes, many. In a way, the Hilbert curve is a is an arbitrary choice. It just has several nice properties and seems to work for this application. I'm very interested in exploring alternative ones. As for the 1D to 3D, this morning I removed one slide from this talk, which I now regret, which is actually a 3D model of these 2D landscapes, where you can sort of much better sort of interpret the, if you will, information content or joint information content across the genome, or in this case, across the single chromosome. I'd be happy to share that offline. I removed it because I am personally not necessarily convinced that we need a three-dimensional solution to look at genomic data, at least not as of yet. I think currently most of our interactions with our data is still on a computer screen. We're not yet, routinely using these devices to look at our data. This might change, but I think it's I don't see enormous value in that. And to speak to Jim Costello's point, I did show a 3D map, kind of as a kind of a cartoon representation. I don't necessarily see that as a way of actually interacting with the data, but the kids would definitely like it. I agree with that. Yeah, and I'm happy to show that at some other point, or next time I'll be around. I'll show it to you. I think Larry Hunter raises his hand.
Yeah, hi, Wouter.
Hi.
I guess I want to push your metaphor a little bit further. And so when in a modern mapping system, I can say, hey, I'm interested in going to Home Depot and the grocery store and to the coffee shop. Give me a route that gets me to each of those places and then back home with the least traffic. And I'm kind of curious because, the genome, when it turns into life, when it's animated in a particular cell type or the like, is full of relationships. It's not about particular genes or regulatory sites in the dynamic aspect of a cell. It's really about how they interact with each other. And so what's your vision of a mapping system that takes into account or shows us or illuminates the relevant parts of those relationships and the sort of pathways through the genome rather than the static landscape?
Yeah. I think, so excellent points. I think there's two parts of this. One is, how do you reflect in these kind of maps? How do you reflect dynamics between different cell types or assays or general data sets? And the second thing I think is important here is, how do you reflect relationships between different loci? So between Home Depot and the grocery store and things like that, and potential routes between those. I think, and correct me if this is not a good way to sort of dissect this question. I think that as for the first part, the dynamics is something you can very easily take into account in a sort of with a machine-assisted backend. Essentially, in fact, that's what's driving this, right? And so you can use at the very least, you can have different information metrics that result in different map landscapes based on, for instance, where are the areas that are most cell type specific versus most constitutive? Where are anomalies in certain cell types of interest versus not, things like that. So you can recolor this landscape in a dynamic way. And so I started calling this sort of conditional genome browsing, where you might say, like, I have a certain prior. I know I'm interested in studying enhancers, and having that piece of information results in sort of recoloring or redrawing the map landscape. And so maybe that addresses your point a little bit. The other part of it is the relationship between loci. Now, that is a very difficult one. That is one that many people have tried to solve. A very good example of this is gene to enhancer linking. There are probably hundreds of groups working on that project, also as part of large consortia. I think that is fundamentally a very interesting problem. the data to do that properly, given the sort of sometimes not super amazing results on that, might be lacking. So I'm personally holding off on that for a little bit. I think there will be a point where there is enough data to say something more about that. But you are right. That is, this would be an ideal, essentially not just a genomic navigation system across loci, but a genomic navigation through pathways, if you will.
So Wouter, oh, sorry, I jumped in front of a hand. Go ahead, Catherine.
No, please go ahead, because I have a very naive question. If there's time for it, we'll get to it. So.
Oh, thanks. So a lot of these data that you're talking about are generated on cell lines and are fairly static. And with ATAC-seq, and we can do that at single cell resolution on essentially on patient samples now. So where do you see this the expansion of these kinds of ideas? Is it more data? Is it applications of your MOPS to clinical situations? How does phenotype come into it? And also related, ENCODE and projects like that had tens, actually probably hundreds of millions of dollars to do this kind of work. What would you think about recommending to people who want to maximize their output from a single set of experiments?
Yeah.
What would be the recommendation these days?
Yeah, excellent question. So as for the first part, actually most of the data, the majority of samples we used for mapping the chromatin accessibility data is primary cells and tissue. But as for the second part, which I think is probably more important to the point you were asking about, what would you recommend people to do with their own data? My very simple answer to that is put it in a wider context. Like, no single experiment should ever be done in isolation anymore. There's no reason for it. I think it's almost naive to do that. If you're doing a single cell ATAC-seq experiment, for instance, you should make use of these resources we have. In fact, we show in the paper and the supplement that it's almost trivial to take any kind of data set that measures chromatin accessibility and essentially incorporate it in our index and automatically essentially get an annotation for the elements in your data set, right? And so I think there's enormous value, as you of course are well aware of, given your work in the area, particularly for RNA-seq data. There's enormous value in putting your own data into a larger context and a wider perspective based on everything else that has ever been measured and made publicly available. So I think my recommendation would be for any kind of data sets going forward, whether it's in consortia or otherwise, is to keep putting it in these larger contexts that we are creating for people.
Okay, I'll ask my very naive question now. So, which kind of comes on the back of that. So, full disclosure, I'm a structural biologist. I don't generate any of these data sets, nor would I know what to do with them. But I think that I kind of browsed through your site. I think it's really cool, and I guess it came down to two naive questions. One is, that you probably mentioned that it just went over my head. But how did you decide on the data that you included in in this kind of analysis? Because I think that there's, and having worked on ENCODE, right, like there's so many variabilities with antibodies and this and that and the other, and like, so how do you vet that? And then I guess it goes to that question of your own individual data set. I don't generate these, but if you were to, and I think you already kind of answered this, how do your tools help people look at their own? But it sounds like you can upload your own data and analyze it in that broader context. Is that correct?
So yes, well, I wouldn't say you can upload it and then just push of a button. There is a large difference between making it possible and operationalizing it. Okay. So this is something we have done in the past for the chromatin state data I presented in the beginning, where people would actually be able to take their ChIP-seq data, upload it, and get back chromatin state calls according to the same model. So I think yes, but yeah, you're right. I kind of answered that in sense like the methods are there for people to do that, right? As for how do you select the data that goes in here, it's extremely important. It's a really good question. all of this, and I cannot stress this enough, all of this relies on tons of work by people who generate good data, but also tons of work of people who know how to process these data in a uniform manner, detect things like batch effects, correct for them, or are willing to throw data out if it doesn't sort of fit the bill. And so how do we select data for these kind of projects? Typically, it's what you can get, what's high enough quality, and what gives you a wide variety of different cell types and different conditions, right? That's sort of how these things are done. And a reason for that is you want to have a high coverage, just because you will maybe be able to find a more sort of broader set of interesting biology, but also because you want to build these sort of generalizable models. You don't want to just build it on immune cells and then expect that you'll be able to seamlessly integrate, neural tube data, for instance. And so that's something that, we have to, I think the power of these sort of large consortia comes from the fact that they are able to do that, right? They're able to put together enough people with enough money, with enough resources to actually get a representative large set of samples and work with those.
So Wouter, I'm Jim Costello over here. Very nice talk. I appreciate the work and all of the stuff that the consortiums have put together. And I'm gonna maybe pick on Kat a little bit here and use her as an example. So we obviously need to reduce the dimensionality to the point where we can try to interpret things. And I'm a big fan of that. I think we absolutely need it. So I hope my question isn't construed as negative, but I'm curious in how you're thinking about the opposite. So how people could potentially be misled by these data and these maps and our preconceptions of how things are visualized versus what's happening at the molecular level.
Yeah, I think one example that, I think one good example of this is, well, first of all, it starts with expecting miracles, right? So it starts with expecting these methods to actually lead to zero information content loss, which is clearly not the case. And so if you have in case of the chromatin state data with Epilogues, for instance, in case you have a truly rare sample and a truly unique locus, for instance, this method is not good. This method will still give you, it'll give you some sort of a consensus or prevailing pattern. It's not going to give you anything that's truly unique to a single biosample unless it's really very, very, very rare, maybe. Right. And so I think there is a lot of work to be done that that addresses that by, for instance, doing things like anomaly detection. Right, and an anomaly detection has a kind of a sort of a negative tone to it, but it doesn't have to be. It could be like, the odd one out could be the interesting one out, right? And so I think there's many opportunities to build on top of this and say like, okay, well, sure, this is the Epilogues representation of this locus. It looks like indeed there is a gene with a promoter and 3 enhancers around it, and maybe a heterochromatin patch further downstream. But wait a minute. Underneath here is actually, there's 3 samples here that are, have a very well-defined genic enhancer in them, and you're missing them or something, right? So I think that and again, this comes back to the idea of genomic navigation systems. I think there's a lot of room for coming up with methods and building tools that really hold people's hands a little bit and don't just give them a summary and like as if that's the solution, because I think you're totally right. It isn't, right? It still requires a certain sense of, intuition and knowledge to interpret even these summarizations. Yeah.
Great. So Kat still has a job. That's good.
Well, I know, but I think it's awesome because, we think a lot on a very single nucleosome level about these epichromatin states, right? And to try to guide more of our biophysical structural analysis, we want to look at something that's relevant. But to be honest, I mean, Jim's totally right to use me as an example in this case. Like when I look at these kinds of analyses, I am not equipped to make a good logical conclusion and say like, oh, great, here's the state I'm going to look at biophysically, and so I think that that developing tools to help very naive people like me navigate that is super important, right? Because otherwise we're looking at stuff that I've totally misinterpreted, which happens a lot, I think, with the bridge between global chromatin and like a single nucleosome.
Right.
Yeah, or think about different visualization techniques that could link your expectation to the data or something along those lines.
Yeah, totally. Yeah.
I see there's one more question. Not sure if this was considered, but are there any striking patterns or differences between data sets from cell lines versus ex vivo cells of the same type? It's a good question. It's hard to answer because a lot of these cell lines are, I mean, they're essentially cancer cell lines. And so, yes, there are going to be many major differences. And so I think it's tricky. So those are certainly differences. So I'm not sure beyond that how to answer that because those effects are so huge.
Cool. Well, with that, we're already past the top of the hour, but just wanted to thank Wouter for giving this talk today. Thanks to everyone who attended as well.
Thanks.
Thanks. It was a really nice talk.
Thank you.